All right, so today we're diving into something that might not sound all that exciting at first, chromatography,
Hmm?
but stick with us because it's actually way more interesting than you might think. And honestly, it underpins so many things we rely on every day, like life-saving medicines, making sure our food and water are safe, even environmental monitoring, it's everywhere.
Yeah, it's like one of those hidden heroes of science, you know? It's this amazing process that can separate mixtures into their individual parts. It's kind of like, think of a smoothie and somehow magically getting all the separate fruits and veggies back out.
Exactly.
Yeah.
And all this magic happens in these tiny little tubes called chromatography columns. They're packed with these special particles. But here's the thing, even though scientists have been using chromatography for ages, packing those columns perfectly is still a bit of a mystery.
Yeah, it really is. And that's what today's deep dive is all about. We're going to be looking at this research paper with a really long title. It's called Evaluation of the Uniformity of Analytical-Size Chromatography Columns, prepared by the downward packing of particulate slurries.
Wow. Yeah, try saying that 3 times fast.
Right.
So who are these brilliant minds behind this research paper?
Well, we have Victor Wong, R. Andrew Shalliker, and Georges Guiochon.
Okay, so what were they trying to figure out with all of this research?
Well, they really wanted to understand if the way a column is packed actually affects how well it works. Like, do different parts of the column perform differently? You know, they suspected that might be the case.
Okay, that makes sense. I mean, it seems logical that different parts of the column might behave differently.
Yeah.
So how did you go about testing this theory?
So they used a pretty clever approach. They started by packing a bunch of columns, each to different length, and then, get this, they carefully cut each column into these 5 centimeter sections.
Wait a minute. They were performing surgery on these tiny metal tubes.
Pretty much. It was a very meticulous process. This sectional analysis was really important because it allowed them to actually isolate the performance of each part of the column.
Oh, that's really clever. So then they could directly compare how each section was performing.
Exactly. They tested each of those little sections to see how well it separated mixtures. And they were looking at things like how efficient the separation was and how much tailing occurred.
Okay, so what did they find? Was there like a clear winner? Like, was the bottom section always the best because it's packed the tightest?
Well, that's where things get interesting. The results were actually pretty surprising. First, they found that the packing density wasn't uniform at all. The bottom section was definitely denser than the top, but it didn't perform as well as you might expect.
Hmm. So tighter packing doesn't necessarily mean better performance.
Exactly. It's more nuanced than that. And what they actually found was that the middle sections of the longer columns, those were the ones that turned out to be the most efficient with the least amount of tailing.
Okay, can we just back up for a second?
Yeah.
What exactly is tailing?
Oh, yeah, good point. So imagine you're separating a mixture of dyes, right? Tailing is when the colors kind of bleed into each other, and it makes it harder to get that nice, clean separation.
Got it. It's like the colors are smudging together instead of staying in their own neat little bands.
Exactly. And that's not what you want in chromatography. You want those distinct peaks.
Right, so the middle sections were giving those nice crisp separations with minimal tailing. What are the top section? How did that perform?
Well, the top section consistently performed the worst, no matter how long the column was. And this really suggests that just cramming those particles in isn't the key to a well-functioning column. There's more to it than that.
Okay, I'm officially intrigued. Why is packing these tiny columns so complicated? What's actually going on inside those tubes that makes it such a challenge?
Well, Wong, Shalliker, and Guiochon, they identified three main forces that are at play during the packing process. You've got particle momentum, pressure, pulsations, and wall friction. And all these forces, they act differently in each region of the column, creating a kind of packing density gradient.
Whoa, okay, hold on. Let's break that down a bit.
Oh.
What exactly is particle momentum?
So think of it like dropping a marble into a jar of sand. The harder you drop it, the deeper it sinks, and the more the sand around it gets packed.
Right. Okay, yeah, I can visualize that.
That's essentially particle momentum, the force with which those particles are hitting the bottom of the column during packing.
So the faster they're moving, the more force they have, and the denser that bottom section gets.
Precisely.
Okay, that makes sense.
Yeah.
But you also mentioned pressure pulsations. What's that all about?
Right. So that has to do with the pump they use to pack the columns. The pump isn't perfectly smooth. It creates these little pressure changes, and those changes can actually affect how the particles settle.
So it's like shaking that jar of sand. Sometimes you get a nice even packing, and other times you get air pockets or voids.
Exactly. And those voids can mess with your separation.
So it's not just about how tightly you pack it, but also how evenly those particles settle.
Okay.
Okay, and then the third force you mentioned was wall friction.
Right. That's the friction created by the particles rubbing against the sides of the column as they're being packed. You might think it's insignificant, but remember we're dealing with really tiny particles in narrow columns.
Right, so even a little bit of friction can make a difference.
Exactly. That friction can have a big impact on how those particles arrange themselves. And it can even create like little clusters of particles that affect the overall packing structure.
So we've got these three forces all acting differently in different parts of the column: particle momentum, pressure, pulsations, and wall friction.
Yeah.
How does that explain why the middle section performs the best?
Well, the middle section, it's kind of like the Goldilocks zone. It's far enough away from the initial chaos of packing at the bottom, where all those particles are hitting with so much force.
Right.
And it's also protected from that direct solvent impact at the top. So the particles in the middle section, they have a chance to settle more consistently without being bombarded by all those extreme forces.
So it's like the particles in the middle have time to find their happy place.
Exactly. It's a calmer environment,
Okay.
and that leads to that sweet spot of performance we were talking about.
Okay, that makes sense. And what about the top and bottom sections? Why do they tend to underperform?
Well, the bottom section or the outlet section, it kind of gets hit with the worst of both worlds. It bears the brunt of that initial particle momentum.
Right, all those particles slamming into the bottom.
Exactly. So it ends up being really dense, but it's also vulnerable to those pressure fluctuations from the pump. Remember those little pressure changes we talked about?
Yeah, like shaking the jar of sand.
Right. And those fluctuations can actually create voids in the packing.
Oh, so even though it's tightly packed, it's not necessarily packed consistently.
Exactly. And those voids, they can really mess with your separation.
Got it. So what about the top section, the inlet section?
Up there, you've got less particle momentum because the particles have traveled further through the column, so they're not hitting with as much force. But you've also got that direct solvent impact.
The solvent's just blasting those particles right as they're trying to settle.
Yeah, basically. And that can cause what's called channeling.
Channeling.
Yeah.
Okay, I'm not familiar with that one. What does that mean?
So imagine you're looking at the packing material. It's supposed to be this nice even bed, right? But channeling creates these little tunnels in the packing. Like imagine a water slide with some parts smoother than others. The water's going to rush through those smooth parts, right?
Yeah, it's going to take the path of least resistance.
Exactly. So channeling is like creating those fast lanes in the column. It makes the separation uneven and inefficient.
Wow, it's amazing how much is going on at such a tiny scale.
I know, right?
Huh.
And it all comes back to those three forces: particle momentum, pressure pulsations, and wall friction.
So the researchers really nailed it when they said that the way a column is packed really does affect how well it works.
Absolutely.
Huh.
It's not just about cramming the particles in there. You have to consider all those forces and how they interact.
And here's something I found really interesting. Wong, Shalliker, and Guiochon, they found that the length of the column actually changes how those forces interact. Like in shorter columns, the outlet section can sometimes be the best performer.
Yeah, that's one of the more counterintuitive findings. It all comes down to the balance of forces. In shorter columns, the impact of wall friction is reduced, and that compression force from the solvent becomes more dominant.
So it's like a tug of war between these different forces, and the length of the column changes who wins.
That's a great way to put it. And that actually leads to a really interesting suggestion from the researchers. If you need a short, high-performing column, it might actually be better to pack a longer one and then carefully cut off the less efficient top and bottom sections.
So you're basically sacrificing those less than ideal sections to get to that sweet spot in the middle.
Exactly. And it highlights the fact that packing these columns, it's really more of an art than a science. There's still a lot we don't fully understand about the process.
You know, before we started this deep dive, I picture those particles just settling neatly into the column like filling a salt shaker. I had no idea. There's so much complexity involved.
Right. It's easy to underestimate the intricacies of something we can't see with the naked eye. But as we've learned, even tiny particles can behave in surprising ways when you throw physics into the mix.
It makes you wonder if there are even better ways to pack these columns that we haven't even thought of yet. What if we could somehow control those forces more precisely?
Now, that's a great question, and one that researchers are actively exploring. There's a lot of potential for innovation in this field, whether it's developing new packing techniques, using different materials, or even coming up with entirely new column designs. It's a fascinating area of research for sure. And, it makes you wonder about those larger scale applications of chromatography. Like, what about in industrial settings? Do those same principles apply?
Yeah, that's a good point.
Yeah.
It's one thing to pack a tiny column in a lab, but what about when you need to pack something the size of like a water tower?
Right. Scaling up the process definitely brings its own set of challenges. It's not just a matter of making everything bigger. You have to really think about how those forces we've been talking about, particle momentum, pressure, pulsations, wall friction, how they all change when you're dealing with huge volumes and faster flow rates.
Yeah, it's like the difference between baking a batch of cookies and running a whole bakery. You can't just use the same recipe and expect everything to work out perfectly.
Exactly. And when you're scaling up, you also have to consider things like cost and efficiency. You can't be spending a fortune just to pack a single column, and you need to make sure the packing process is still consistent and reliable.
It makes you appreciate the expertise of the engineers and scientists who work on these large-scale chromatography systems. They're basically conducting a massive symphony of particles, fluids, and forces.
It's true. They have to have a really deep understanding of all the factors involved. And the stakes are high, especially in industries like pharmaceuticals, where the purity of a drug can literally be a matter of life and death.
You know, this deep dive has really given me a new perspective on chromatography. It's one of those things we don't often think about, but it plays such a crucial role in so many aspects of our lives.
It's kind of like the unsung hero of modern science. It's quietly working behind the scenes to make sure our medications are effective, our water is clean, our food is safe. It's pretty amazing when you think about it.
Absolutely. And it's all thanks to those tiny packed particles and the intricate dance they perform to separate mixtures and reveal the hidden secrets of our world. So the next time you hear the word chromatography, I hope you'll remember all the complexity and fascinating science that goes into it.
I hope so too. It's a reminder that even the smallest things can have a huge impact, and that there's always more to discover.
Well said. And on that note, I think it's time to wrap up this deep dive. Thanks for joining us today. It's been a pleasure exploring the world of chromatography with you.
The pleasure was all mine.
And to all our listeners out there, stay curious, keep exploring, and we'll catch you next time on the deep dive.